And good afternoon, esteemed colleagues and researchers. It's a pleasure to be with you today to delve into an emerging frontier in neuroscience, photobiomodulation, and its multifactorial effect on the central nerve system. As you know, this is not merely an experimental curiosity. It represents a promising road for understanding and potentially treating a range of neurological and psychiatric disorders. As you know, photobiomodulation, once known by many names, cold laser, soft laser, low-level laser therapy, and has evolved significantly since Mester's early experiments in 1964 and its first clinical application in 1974. As you know, photobiomodulation, a groundbreaking non-invasive therapy using red near-infrared light to modulate cellular and molecular processes in the nervous system. Its application in neuroscience span neurodegeneration, traumatic brain injury, as Dr. Norbert said, and cognitive enhancement, offering novel insights into brain repair and function. What makes this fascinating is how light, something external, can influence intracellular mechanism. As you know, in the body and in organs, we have biological tissue are replete with the photoreceptors, such as cytochrome oxidase C, as you know, the primary mitochondrial chromophore. And also we have many number of hemoglobin, myoglobin, and melanin. Each absorb a specific wavelengths and translate this energy into cellular responses. So mitochondria are the energy epicenter of neuron, and dysfunction in mitochondrial pathway is increasingly implicated in neurodegenerative and psychiatric disorders. As you know, in the pathology of many number of neurodegenerative disease, we can see mitochondriopathy. So PBM or photobiomodulation targets cytochrome C oxidase in the mitochondrial respiratory chain, as you see in the diagram. Upon light absorption, this enzyme accelerated oxidative phosphorylation, boosted oxygen consumption, and significantly increased ATP production. So why does this matter? Because neurons are highly energy demanding cells. Their membrane polarization and synaptic activity consume vast amounts of ATP. By restoring mitochondrial function, photobiomodulation helps stabilize neural activity and prevent cell death, particularly under condition of stress, ischemia, or oxidative damage. So PBM targets key neural cell types, including neurons, astrocytes, and microglia. That is driving a reparative and protective effect in this type of cells. So neurogenesis and synaptic plasticity, it's another, effects of photobiomodulation. On the other hand, photobiomodulation enhances neural proliferation and differentiation that is critical for recovery after brain injury. Studies show it promotes axonal regeneration and synaptic connectivity, aiding functional recovery in models of stroke or traumatic brain injuries. So in targeted cells, for example, glial cell modulation and microglia and astrocytes central to neuroinflammation are regulated by photobiomodulation. It shifts macroglia from pro-inflammatory M1 to anti-inflammatory M2 phenotypes, reducing neuroinflammation. Astrocytes under photobiomodulation exhibit improved mitochondrial function, supporting neural metabolism. Sorry, can you hear me? Okay. As you see in this diagram, photobiomodulation using red or near-infrared light penetrated the cell membrane and is absorbed by mitochondrial chromophores, particularly cytochrome C oxidase, as I said. This interaction in step 1, as you see, step 1, boosts mitochondrial ATP production. Step 2, which is critical for energy-intensive neural function. As ITP level rise in step 3, extracellular ATP start to signals to two primary cholinergic receptors. As you see in the slides, you can see here, we have P2X receptors and P2Y receptors, which are ion channel P2 receptors, P2X receptors, sorry, which are ion channel that allow calcium influx. Step 4. And P2Y receptors, which are G protein coupled and activate the phospholipase C, inositol triphosphate, D-acylglycerol pathways. Step 5. As also loading to calcium release from the endoplasmic reticulum. Now, here, where things get interesting. This rise in intracellular calcium activates several downstream pathways, as you see here in the diagram. One major route involves protein kinase C and the ERK pathway, which collectively enhance cell survival. Another path activates calmodulin, calcium binding protein, and PI3K and AKT pathway will establish anti-apoptotic signaling axis, as you see in step 6. Simultaneously, photobiomodulation suppresses pro-apoptotic mechanism, effectively blocking cell death pathway and promoting cellular resilience. In summary, we can say photobiomodulation orchestrated a highly coordinated symphony of bioenergetic, calcium-dependent, and also anti-apoptotic pathway, which together created a neuroprotective effect in environment. This mechanism not only highlights the biological death of light-based therapy, but also its therapeutic potential in neurodegenerative and neuroinflammatory conditions. Okay, you can see in the next diagram. Let's take a moment to explore how photobiomodulation activates a network of signaling pathway, the convergence on cell survival, anti-apoptotic activity, and inflammation controls. As you see in this diagram, we have 2 and 3 important pathways. It begins when photobiomodulation through red or near-infrared lights stimulates the mitochondria, step 1. This leads to boost in ATP production, step 2. A critical energy resource for neural activity. Some of this ATP is used to activate adenyl cyclase, which catalyze the formation of cyclic AMP, CAMP. Now, CAMP is not just byproduct, it's second messenger. It activates protein kinase A, which trigger 2 major protective routes. Protein kinase A activate CIRT1 signaling pathway, leading to an increase in the heat shock proteins. As you know, heat shock proteins, it's very important type of protein that promotes cell that can combat cellular stress and inhibit inflammation. Heat shock protein signaling. In this case, it's very important. It's another effect of the biomodulation. Protein kinase A also activates RAS, which sequentially stimulates ERK pathway, ultimately lead to enhanced cell survival. As you know, in the cellular mechanism, we have many number of signaling pathway. After activation of CAMP signal transduction, maybe promote actually synthesis of many number of enzymes. For example, protein kinase A and protein kinase C. Two important promoter for cell survival. Simultaneously, mitochondria under photobiomodulation may generate a moderate level of reactive oxygen species, ROS. You are familiar with ROS. Not damaging in this context, but rather signaling molecule. ROS activates SRC kinase, which promotes both phagocytosis and PI3K AKT signaling. The latter being a well-known anti-apoptotic pathway. Through this cascade, photobiomodulation doesn't just keep the cell alive. It orchestrates a survival program, balancing energy production, stress response, inflammation, and resolution and cellular repair, as you see in the cascade. In short, this diagram reflects how photobiomodulation taps into the cell on-regulatory machinery, turning light into signals for neuroprotection, recovery, and resilience. Okay. In this slide, and also you can see the remarkable cascade of brain repair mechanism initiated by the activation of genes, as Dr. Nurbakhsh said. When genes such as NF-kappa are activated, they act as master switch, turning on numerous downstream genes. This process sets off a chain of events critical to healing and regeneration. Key among these are growth factors. We're familiar with the number of, growth factors, for example, BDNF, brain-derived neurotrophic factor, and NGF, molecules that act as nature's engineers, stimulating tissue growth, repair, and the survival of the neurons. This factor paved the way for neuron repair, ensuring damaged cells are restored, and new synapses are formed. Improving communication may be actually maybe happened in this some networks within the brain. Additionally, activated genes spark stem cell activation, unlocking the potential of generation of new cells, while carefully balanced inflammatory mediators help clear debris and prepare the environment for recovery. So this balance is very, very important. Central to all of this process is BDNF, a cornerstone of neuroplasticity and repair. Together, this mechanism illustrates the brain's extraordinary capacity to heal, adapt, and renew itself. Another remarkable outcome of photobiomodulation is its effect in cerebral blood flow. Impaired perfusion is hallmark of many brain disorders. Enhanced cytochrome C oxidase activity, which trigger the release of nitric oxide, a potent vasodilator. Nitric oxide, as you know, is a small lipid soluble gas with a short half-life. It was the first gas discovered to act as a neurotransmitter, freely diffuse across membrane to modulate intracellular enzyme rather than surface receptors. Okay, we can see photobiomodulation induced nitric oxide. Nitric oxide has many important effects: expands blood vessels, restore oxygen supply, support neurotransmission and neurogenesis, as you see here. Involved in process as varied as mood regulation, memory, neurodegeneration, making it a key player in photobiomodulation systemic benefits. I mean, releasing and synthesis of nitric oxides. Okay, after this important component, we have another important component as a calcium. You are familiar with the calcium and the role of calcium in central nervous system. Photobiomodulation modulates calcium signaling by influencing voltage-gated calcium channels, affecting inositol triphosphate mediate pathways, supporting homeostasis, and this modulation has profound downstream effects, including neuroprotection, synaptic plasticity, gene expression changes. So, yet, calcium dysregulation contributes to many number of pathological conditions, for example, in Alzheimer's disease, in ischemia and seizure, aging-related dysfunction. So calcium actually is very important homeostatic factors. So after photobiomodulation, we have releasing of calcium and modulation of calcium release and calcium storage in the bodies. By restoring of mitochondrial membrane potential and reducing of ROS generation, photobiomodulation offers a shield against oxidative stress, a major cause of DNA, protein, and lipid damage in neurons. After photobiomodulation, for some session, some studies show that we have promoting of neurotrophin expression, such as BDNF, glial driving neurotrophic factors, GDNF, NGF, and this cascade enhance actually neurogenesis, synaptogenesis, functional recovery following neural injury. This effect highlights photobiomodulation's potential role not just in neuroprotection, but also in neuroregeneration. Beyond the cellular level, photobiomodulation can recalibrate intrinsic brain networks. For example, it affects excitation inhibition, ATP-driven neurotransmitter release, e.g., rhythm balance, and potentially restoring coherence in disorder like ADHD, depression, and Alzheimer's. So, some recent work even suggests that photobiomodulation may modulate reaction time and cognitive flexibility in healthy subjects, opening doors for its use in enhancement applications. So, in summary, photobiomodulation represents a paradigm shift in our understanding of how light can influence the brain at a cellular and molecular level. By enhancing mitochondrial function, modulating inflammation, promoting neuroplasticity, and supporting neurogenesis, photobiomodulation offers a powerful, non-invasive tool for both basic research and clinical application in neuroscience. As we stand at the edge of new era in neuromodulation, these 5 emerging areas promise to expand the boundaries of what photobiomodulation can achieve, not just as a therapy, but as a platform for understanding and enhancing brain function. Let us continue to explore, innovate, and illuminate the path forward, quite literally with light. Here we can see a precision transcranial for neurodegenerative disorders. And also another trend is personalized wavelengths and dosing protocol to target specific brain regions in Alzheimer's and ALS dysfunction, amyotrophic lateral sclerosis. And also another trend is optogenetic, like neuromodulation with photobiomodulation, using light to non-invasively control neural circuits, merging photobiomodulation with optogenetic for precise behavioral modulation. Another important trend for research is include of photobiomodulation and gut-brain axis investigation, systemic photobiomodulation effect in gut microbiota, and its bidirectional influence in neuroinflammation and mood disorder. And also another important and interestingly subject is maybe nanoparticle enhanced photobiomodulation delivery. For example, gold nanoparticles conjugated with light sensitive agent to amplify photobiomodulation efficacy in deep brain structures. And finally, we can say photomodulation has many effects in neuroimmunology, exploring photobiomodulation's role in autoimmune conditions like multiple sclerosis, focusing on T cell and glial cell crosstalk. So I think that we can start many number of projects in this case, and we can, have a collaboration by each other for, designing many number of mega projects for these trended cases. So in summary, I'm here for having your, if you have any comment or if you have any question, you can ask and please share with us. And thank you for your attention. Okay, if you have any questions.
Thank you very much. Mehrabi. Thank you very much for your excellent presentation, very useful. Any question?
Are there any research? Thank you, Ms. Mehrabi. Are there any research about multiple sclerosis or amyotrophic lateral sclerosis, kinds of these problems, neurological severe problems?
Yeah, actually, in ALS or amyotrophic lateral sclerosis, we have some complications, spasticity, as you know, rigidity, and also mental illness, and so motor neuron dysfunction. So in these cases, we have another treatment, or we have not any gold standard type of treatment. So actually, it's just combination therapy with another, interventions. For example, we have rehabilitation, physical rehabilitation, and in the best side of physical rehabilitation, we can use from photobiomodulation centrally or peripherally. Actually, one of my projects for PhD student thesis is about of efficacy and safety as photobiomodulation in these cases. I mean, in amyotrophic lateral sclerosis patients. So we can try to explore efficacy or safety as these devices in this case. I very hope we can see any efficacy in this case. But we have many complications, but we will try to do.
Sure. We are.
Can I ask my question, please?
Sure.
All right. Okay, from Nigeria. This is Lawrence Adedayo from Nigeria. Thank you for the wonderful presentation, and thank you for enlightening us on photobiomodulation therapy. My question is this: I want to know if we can use that PBM on animal models of brain. If you have actually used them in those things for the treatment of Alzheimer's disease. Alzheimer's disease, they have to potentiate it from animal models of Alzheimer's disease, and of course, for neuropathic pain. I don't know, maybe you have applied them. If you have applied them, I would like you to expatiate on the mechanisms of how you can use this in treatment of these animal models of disease. Thank you.
Okay, thank you for your question. Actually, in this some pathological condition, actually in inflammatory pain or neurological pain, we have, accumulation of cytokines and many number of inflammatory factors. So one of the facts about the photomodulation is improvement of, decreasing of level of inflammatory markers, arterial markers. So after applying the photobiomodulation transcranially or peripherally or intranasally, some studies show that we have decreasing of level of inflammatory markers. So, for example, interleukins, interferons, and something like that. So in this case, one strategy back today, decreasing level of cytokines, inflammatory cytokines, and another back to the decreasing the level of oxidative stress in the body, because in a painful condition we have accumulation of many number of components, for example, hydrogen acide, substance P, histamine, serotonin, and something like that in the painful body or after damaging the body. So during a photobiomodulation application, we have, a scavenging effect, So the one theory back to the scavenging effect of photobiomodulation, and another back to the decreasing level of inflammatory component and factors.
Thank you. Thank you so much for your response. And lastly, I don't know, maybe you have a workshop training hands-on, because we are not actually exposed to the the lower laser photo, because I noticed from your presentation, the lower laser is the one that is actually effective, not because it has less side effect. So my question is that I don't know, maybe you can have hands-on training and probably invite people to your lab so that they can learn some of these things, and we go into collaborations over time, because this technique is new and a lot of people in sub-Saharan Africa might be interested in learning the protocol and the procedure. We don't actually, people are not using it so much in sub-Saharan Africa where I stay currently. So if you have hands-on training, we'll be glad to come and then learn one or two techniques and replicate the process here, and of course open up for collaboration in subsequent time. Thank you.
Great, fantastic. It's my pleasure. We can have a collaboration and many number of workshops together in collaboration.
Sure, thank you for your.
Hello, Dr. Soraya. I have a question.
Hi, thank you.
This is Dr. Najia from Pakistan. I am using photobiomodulation therapy in my dental patients for past few years, and I basically treat patients with myofascial pain related to head and neck, especially in myalgia and muscle cramps, and it's been effective so far. My question is, can we use this device, like the one I have? It has a low level range of 400 and 635 nanometer. What device I'm using? It's a diode laser. And can we use this specific wavelength from a diode laser, a low level laser photobiomodulation therapy for migraine? And how do we do that? Is there any specific points that we have to use it? Because I frequently see patients with tension headaches, migraine headaches. So how about that? What is your insight about that?
Actually, thank you for asking this question. I think that Dr. Salehpour and other colleagues can answer to this question, because actually we have 4 lecturers. And the next lecturer, my colleagues, will be explained about some clinical approach of photobiomodulation in neuropsychiatric condition, and then after that, Dr. Salehpour can be complete this discussion. But we can say in the many number of studies, the effect of photobiomodulation in a neurological condition or neuralgic pain, and also in some and other cases, hyperalgesia, allodynia in animal study or clinical trial studies. So please, let's continue the discussion with the next colleagues, and after that we get back to the question. Thank you for
Thank you.
Thank you very much.